Another micronutrient that is interesting, iron. Okay, so the hypothesis came about from John Martin, an American oceanographer. So it is known that there are presence of high nutrient, low chlorophyll regions in the open ocean. So it doesn't make sense. If you have nutrients, if you already have your N, you already have your P, you already have your nitrogen, you already have your phosphorus, why is the chlorophyll still low? Okay, so the proposed hypothesis is the proposed hypothesis was that there is another limiting factor that is constraining the development of primary producers. So he proposed that iron is the limiting factor. So during one of the scientific conferences, he made this very outrageous statement: give me half a tanker of iron and I will give you an ice age. Do you understand what it means? Or why did he say this?
Maybe because the iron and micronutrients actually encourage the growth of the algae, leads to maybe algal bloom, and then it affects a lot to the ocean and leads to ice age.
Oh, so when you so since the hypothesis states that iron is the one that is limiting the primary production in the seas, in the open ocean, if you put a lot of iron into the oceans, the primary producers will bloom. When the primary producers grow, they'll fix a lot of CO2. There'll be a drawdown of CO2 into the oceans. That means the CO2 in the atmosphere will drop. Okay. And since you've seen from the earlier lecture how the concentration of CO2 in the atmosphere is related to the global temperatures, with a decrease in the CO2 in the atmosphere, it is hypothesized that that will bring about the next ice age. Okay, temperatures will drop. Okay, so that's why he made this outrageous statement. So during the presentation, he was presenting uh, preliminary results. Okay, what he did was he took open ocean waters, he fertilized them with, which means he enriched the waters with iron, and he found out that phyto the the chlorophyll, the primary producers increased a lot. So he was very confident. He was very happy with the results. The addition of iron into the waters showed that the uh, primary producers grow tremendously. That shows that yes, the primary producers are limited by iron in the open oceans. Not all oceans, okay? Only selected ones, for example, in the Southern Ocean and also in the North Pacific Ocean. Okay, only certain areas you see the phenomena where you have high nutrient, low chlorophyll region. Okay, so of course, with all new with with all new ideas, there are lots of resistance, a lot of naysayers. Okay, people who don't agree with him. So the rebuttal was he was working with Flask. Okay, he was working with at a scale of flask. So they said they state that maybe the flask was it's too small. The scale is too small, cannot be related to the open ocean, because if you remember from earlier lectures also, when you talk about the growth of microorganisms, the surface to volume ratio is important. So when you place microorganisms in a smaller container, somehow the the surface over the area over volume ratio increases, providing more space, more area for the microorganisms to obtain nutrients and stimulate the microorganisms. So by placing the uh, seawater in flasks, smaller flasks, that is already stimulating the microorganisms within the flask. That is how they debated with him, stating that at a scale so small, you cannot really tell whether it is the iron or whether it is the bottle experiment that is causing the change. Okay. So another thing is, when you're working with iron, these are micronutrients. So everything, the procedure has to follow very clean approach. Okay, you have to use clean methods. So as I mentioned, usually when you're working with micronutrients, you will use go flow, you will use Kevlar lines, you will use clean rooms. Okay, you have to use all this equipment in order to minimize contamination from dust, because dust can contain iron. Okay, that's why uh, primary concern was contamination and also the scale of the experiments, the bottle experiments itself. So as with all scientists who believed, really truly believed in the idea, he went about applying for a big grant to start off a cruise. Okay, they called it the iron fertilization cruise, IronX cruise, iron fertilized, where they proposed to carry out iron fertilization experiments. Okay, in the Pacific Ocean. So he obtained the money, okay, a few million US dollar, but sadly he passed away before the cruise could be carried out. Okay, so however, his collaborators and co-PIs decided to continue the cruise in his honor. So they did the experiment. They fertilized the ocean, an area of the ocean, and they used a certain tracer to trace the spread of the iron, and then they collected, and then they observed after a few days that it's true. The um, phytoplankton increased tremendously, significantly. So it shows then that it is true that it is the iron that is limiting the phytoplankton in the open oceans. Okay, so if you add iron to it, you can get a phytoplankton bloom and increase in primary production. But does that mean there will be a net drawdown of CO2? What does the net drawdown mean? A net drawdown of CO2 means when the phytoplankton grows or blooms quickly, you know that it is absorbing or fixing CO2 from the atmosphere. Will this will this cause a net drawdown, which means will this cause an actual decrease in the CO2 in the atmosphere? Okay, so more and more experiments were carried out, not only by Americans, the Japanese also did it. Uh, I know of the Japanese, but whether the Europeans did it or not, I think they did it also. So other regions, other countries, they were doing these iron fertilization experiments. And what the Japanese found out was, yes, there is drawdown, but the net drawdown is insignificant because whatever that is being produced by the phytoplankton, whatever CO2 that is being captured by the phytoplankton, is also released by the zooplankton, by the bacteria. So the heterotrophs are also working, opposing the carbon fixation of the phytoplankton. So you may have an input of CO2 by phytoplankton, but you will have an increased output of CO2 by the heterotrophs. Okay, so it is not so straightforward, it is not so easy. Just by fertilizing the open oceans with iron might not bring a net drawdown of CO2. So it will not solve uh, the problem of global warming due to increasing CO2. Okay, so it's back to the drawing board. Still a lot more things to work out before uh, this kind of experiments can be applied. Okay, so it's not so straightforward. Okay, so yeah, so this is the drawdown of the CO2. However, the bacteria will also release CO2. So the net drawdown is in question now. Okay. So in relation to climate change, sulfur became important, sulfur cycle, when they realized that phytoplankton produces a major osmoregulatory solute, okay, a solution. So these phytoplanktons usually will secrete DMSP, dimethyl sulfonyl propionate, okay, as it carries out photosynthesis. So uh, this is a solution that is released by the phytoplankton as it carries out its photosynthesis. Okay, so this DMSP, when it's released in the euphoric zone, is degraded by bacteria into DMS, dimethyl sulfide. Okay, in this structure. Then in the euphoric zone, with the UV ray sunlight, the DMS breaks down into methane sulfonate, sulfur dioxide, and sulfate. Okay, this sulfate will vaporize into cloud condensation nuclei, CCNs, forming low-hanging clouds. Okay, so this is the one that can catalyze the formation of clouds. So when you have clouds, it will cool the surface of the earth. Okay, so then people begin to realize, hey, maybe this can solve our global warming problem. Okay, so the thinking is maybe an increase in the CCN concentration can counteract an equivalent increase in CO2 concentration. So that's what scientists began thinking of. Okay, so this shows the uh, diagram where the phytoplankton will produce the DMSP, which will be degraded by bacteria into DMS. So this is the structure of the dimethyl sulfide, which will be further broken down by UV light into this, and this will become your cloud condensation nuclei, forming cloud to cool the earth. So it became more interesting when they found out that tropical phytoplankton communities are more efficient producers of DMSP. So people began to have an idea that when the oceans become warmer, the geographical spread of tropical phytoplankton communities will increase. So there will be more areas where the tropical phytoplankton community can grow. So when more tropical phytoplankton grow, that may increase the CCN, increase the clouds, and reduce temperature. Okay, so the Gaia hypothesis is based on the notion that Mother Earth is alive, okay, and that changes that we make to Earth, extreme changes, can be reverted back through the action of Earth itself. Okay, so there's a feedback mechanism from the Earth to maintain homeostasis, to maintain a, 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 a suitable environment for organisms. Okay, so as temperatures increase, more tropical phytoplankton, more CCNs, and the Earth will cool. The problem is the correction might not be fast enough to ensure human survival. Okay, so that is why it's just this is just something that scientists usually write in order, to make their ideas more interesting. Okay, but a lot of times it is not yet practical.